Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Colorants in Devices, Integrated Strategies Toward Regulatory Approval. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for up to 90 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using the chat panel. And at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically. Please note that this event will be recorded and made available to you for future download. And at this point, I would like to thank Toxicon, who has helped develop the content for this presentation. Toxicon has been working with the pharmaceutical, biotechnology, and medical device industry for nearly 40 years. Toxicon is a contract research organization providing in vivo analytical and in vitro testing services. Safety services include toxicology, pharmacokinetics, toxicokinetics, bioavailability, ADME, chemical characterization, impurities analysis and synthesis, bioanalytical, and microbiology. The company currently has approximately 80 animal rooms and has extensive and fully equipped surgical suites, along with support services that include efficacy and research services and histology and clinical pathology. The company currently serves over 1,000 active customers across the pharmaceutical, biotechnology, and medical device sectors throughout the world. Another thanks goes to Arazi Group. Arazi Group Consultants is a global, multifaceted regulatory affairs firm that provides all types of medical device manufacturers with client-specific solutions throughout the product life cycle, from early development to post-market activities. With over 19 years of expertise, their experienced consultants are responsible for the successful registration of thousands of medical devices in 100 countries worldwide. Arazi Group is based in Vancouver, British Columbia, Canada. Their flagship product, LicenseSale.com, is a new, simple solution for registering medical devices globally. And now I would like to introduce our first speaker for today's event. Mark Thompson is a business consultant and sales and marketing expert with an extensive record of achievement within the global medical technology and healthcare services industry. Since 2013, Mark has been acting as North American Sales and Marketing Manager for Arazi Group Consultants, helping medical and IVD device manufacturers around the world to successfully launch their products in multiple markets through LicenseSale.com. Our second speaker for today is John Iannone, Program Manager, Extractables and Leachables and Special Studies for Toxicon Corporation. Since joining Toxicon in 2004, John has assisted numerous drug and medical device developers with the development of product safety testing strategies. His areas of expertise include biocompatibility testing of combination devices, material and chemical characterization, and study design for custom microbiological and extractable leachable studies. John participates in the development of regulatory guidance such as ISO 10993. Additional responsibilities focus on providing technical support to the Toxicon business development team related to ensuring clients have visibility into Toxicon's capabilities and services, as well as interfacing with the clients regarding unique testing requirements to facilitate the attainment of sound technical risk assessments for medical devices. And now, without further ado, I would like to hand the mic over to Mark Thompson. Mark, you can begin your presentation when ready.
Thank you. Well, good morning, everyone. My name is Mark Thompson, and I am the North American Sales Manager for the Arazi Group. We are an international consultancy spanning several continents and we assist medical device companies with regulatory compliance submissions as they enter new markets. Our company was established almost 2 decades ago, and we now provide services in over 100 countries around the globe. In 2013, our company formed a successful partnership with Toxicon, one of the world's premier accredited research and testing facilities. Both leaders in their field, Arazi Group and Toxicon offer a comprehensive suite of preclinical and regulatory compliance services, helping manufacturers around the world to achieve regulatory approval for devices of all risk classes. Today, we will share our joint experience and expertise with you to shed some light on regulatory strategies and with a focus on colorants in devices. Please feel free to type your questions in the question box throughout the presentation, and I will discuss them during the question period at the end of this webinar. We realize that we have a very diverse audience today, and we have worked to ensure that overall both parts of the presentation will have value for everyone, and we thank you for joining us. When preparing a successful dossier for regulatory approval, colorants are one of a number of elements that are considered prior to the submission. And to ensure submission success, it's essential that there is an efficient and cost-effective process in place for its completion. So before John Iannone from Toxicon talks more specifically about the regulatory challenges of colorants in medical devices, let's start with a look at the bigger picture, the process and strategy behind regulatory submissions in a globalized regulatory landscape. At the Arazi Group, we've been serving early startups, medium-sized, and top 100 multinational medtech companies. And the greatest lesson we have learned through our experience is that the future of a medical or diagnostic device manufacturer depends not only on their ability to innovate, but also very much on the ability to get products to market. And the key to getting your product to market is an integrated strategy toward regulatory approval. Without regulatory approval, no device can be sold, no revenue can be generated, and no growth can be obtained. In spite of this, the practice of regulatory affairs has hardly changed over the last 40 years. It remains a fragmented, costly, and inefficient process managed manually and mostly through local distributors or consulting services. So let's look at the current situation. As growth in the medical device sector continues to shift from the traditional US, EU, and Japanese markets towards emerging markets, and the need to get to market first is becoming more and more important, the regulatory landscape has become increasingly complex. In fact, even in the EU, where requirements should have been harmonized under one directive, the implementation of regulations is rarely uniform across the board. There is more and more pressure on regulatory professionals to keep up with the changing global market, and the industry is looking for new ways to deal with the challenges posed specifically by those emerging markets. Latin America, the Middle East, Russia, India, East Asia. These are the regions manufacturers target today in order to secure their growth. And yes, these markets promise growth, but they also come with their own set of problems. Korea, China, and Russia, for example, require local testing for electrical and toxicity compliance, among others, which may cause undue delay and expense. And there are many more challenges in the new regulatory landscape. As you enter emerging markets, you will find that you may not have all the regulatory and market intelligence that you need for your submission. You may not have all the people and infrastructure you need in the local markets. Overly complex processes and requirements often need high investments. Regulatory barriers and unexpected difficulties delay your time to market. You lose control of your licenses, which are now owned by local distributors. And in that process, you also lose control of your intellectual property. Language and cultural barriers don't make your work any easier. And finally, keeping track of post-market activities, changing regulatory requirements, and license renewals extend the difficulties far past the actual approval process. Let's take a quick poll to see which challenges are most relevant for you.
Great. Well, thank you, Mark, for that. We certainly do have a poll question here for the audience, and you can vote on this in real time by clicking on your screen. The question we have for you, when entering a new market, what poses the greatest challenge to you? And your options on the screen are lack of regulatory and market intelligence. Another option, lack of local partners and suppliers. Your third option, exceedingly high cost. Your last two options, long time to market and license ownership and control of IP. Again, that question you can vote on in real time by clicking on your screen. That question again, when entering a new market, what poses the greatest challenge to you? Looks like most of you have voted at this point, so I'm going to close polling now and share the results with everyone. 44% of you said lack of regulatory and market intelligence. That was followed at 29%, long time to market. 11% of you said license ownership and control of IP. That was followed by exceedingly high cost at 9%, and 6% of you said lack of local partners and suppliers. And with that, Mark, I'll hand back the mic to you.
Thank you, Andrew. Thank you for the polling results. These are all common problems, of course, that we see when working with our clients around the globe. And certainly lack of information is one of the biggest issues that most manufacturers face. With license sale, we overcome that challenge quite readily and are able to help manufacturers seek approvals in a very timely and efficient fashion. Let's return to the slides. Now, if we multiply these and other problems with however many new markets you may be planning to enter with your device, and you can imagine the implications for your regulatory department overall. New challenges require new solutions, and as so often true, technology has the answer. In an increasingly complex and expanding regulatory landscape, you'll need a solution that is available anywhere, anytime, and instantly connects you to all the regulatory and market intelligence, all the services, and all the suppliers you need to remain competitive in a globalized medtech market. The solution is the cloud. Cloud computing is a tremendously powerful tool that brings global registration to your fingertips. Think of it as a network that connects manufacturers, regulatory authorities, experts, testing facilities like Toxicon, and suppliers around the world 24/7. A space that contains all regulatory and market intelligence that you need for a successful application and that connects you with all the people required in the process. The power of the cloud can open up new markets and give your company the decisive advantage of getting there faster and at a more accessible cost. But most important of all, it makes your work easier and it gives you more control of every step in the process. At the Eraza Group, we saw huge potential here, and we developed a tool to harness the power of the cloud for our own operations, to improve our own internal processes, and to win decisive advantage over our own competition. But we soon realized that what we had conceived as an internal solution was really the answer to a much bigger question. How do we maintain control in an increasingly complex and demanding regulatory space? And after 3 years of development and testing, we launched LicenseSale.com. LicenseSale.com is a unique, cloud-based global product registration system that streamlines, simplifies, and expedites the approval process of all types of medtech devices. It is a reliable tool that seamlessly integrates with your company's go-to-market strategy and gives you full control of your regulatory submissions worldwide with real-time updates and instant access 24/7. And as with any cloud-based service, there is no need to buy software or invest in infrastructure. With LicenseSale.com, we effectively reduced both time to market and overall license acquisition costs for a medical or in vitro diagnostic device by as much as 50%. And how do we do that? It is through the efficiencies gained within the system, through the elimination of redundant tasks, and through expert consultation. So to what extent does our system increase your efficiency and productivity as regulatory professionals? Well, let me show you an example. In late 2012, we were approached by global radiation therapy giant Elekta to help obtain market clearances for a wide range of products ranging from simple patient positioning devices to high-risk gamma radiation therapeutics. As a general rule, traditionally, a one family of products, one country application takes about 4 to 6 months to receive approval. using licensesale.com, Electa received approval for 78 different products in 16 different countries. And this was all done in less than 1 year. And here is an example of how licensesale.com helped one of our clients overcome a difficult challenge involving colorants. A material supplier for a Class 3 implant changed their compounding and added blue colorant to the polymeric blend being used in the implants. Class 3 clearance had just been issued for a 510K product when the material supplier modified the polymer compounding agent by introducing blue colorant. Typically, a long and expensive process would be required to make the modification to the product. This may include leaching studies, risk assessment of all elemental leaching, material characterization, and an extensive retesting of biocompatibility. Such a program could take 6 months and probably would cost about $85,000. But because the client had just received 510K clearance on the device through licensesale.com, we were able to submit a special 510K using existing documentation in licensesale.com and testing data from the material supplier, including toxicology and dilution data, which allowed us to make the modification within 30 days. This not only helped prevent substantial delays to the U.S. market by eliminating the need for retesting, but it also saved the client $85,000. Licensesale.com is powered by GRIMS, our proprietary global regulatory intelligence management system, which allows simultaneous registration of multiple products in multiple countries. The GRIMS data management system builds a comprehensive compliance profile per product that takes advantage of previously supplied information to eliminate unnecessary duplication of work when taking the product registration from one market to the other. When preparing a new submission, the system automatically populates new applications with relevant data from your master file and guides you through the requirements of a particular market step by step. The master file is set up when you build your first submission. Licensesale.com eliminates the need to re-upload already existing information and flags what is yet to be provided. And the more you use the system, the more efficient your process becomes. Once an application has been initiated through licensesale.com, GRIMS automatically engages our global network of regulatory affairs experts, connecting the most qualified expert for every facet of your project. This means, for example, if your submission involves colorants, you will have access to professionals with highly specialized niche expertise in that field who will be able to give you all the information you need and help expedite your submission. And when the review process is completed, GRIMS generates the final dossier, which is then submitted to the local authorities. The result is a license owned by the manufacturer and not a local distributor. So how can all of this help you in your regulatory responsibilities? You'll be able to set up and easily control multiple applications in multiple markets at the same time. You will know all the requirements for your applications, and they will always be up to date. You can easily upload required documents into the cloud, and you will have to do it only the one time. Once they're in the system, it will automatically reuse them for your next application. You will be able to access and monitor your submissions online 24/7. The status of all documents and applications is constantly updated in real time. All your documents are reviewed by highly specialized niche experts, and you can consult them anytime. You can submit your applications with assistance in language requirements and legal notarization, etc., often some of the most costly elements of the application process. Working in the cloud with licensesale.com means that all your information is in one place. This includes all your documents, but also your sensitive intellectual property. This is what an application process looks like in the system. You see all the documents that are required and the actions that are pending at a glance. This is the list of documents that are required for your application. Here you see the required language versions and whether you need legalization for a certain document or not. These are the documents provided by you. For each document required, you can either upload them from your computer or select them from the online GRIMS database. Alongside each document on the list, a status icon will appear. The amber pencil tells you that client review is required. If you want to know more about a specific document, click on its title to get a description of the document or to download a copy. The system can be set up to allow either one or multiple people within the organization to upload and manage documents. This means the documents supplied by the R&D department can be handled directly by R&D staff, while RA staff handles documents supplied by their department. When all your documents are complete, you're ready to submit your application as shown by the green check marks. After the submission, you'll be able to view the status of the application and receive updates. You'll have improved communication with the authorities, and in case of questions or issues with the document, you will have all the support you need to address or resolve the issue quickly and efficiently. You can reuse all the documentation easily for subsequent applications. You will be notified in advance of license expiration and provided with the tools for timely renewals. The rapid industry adaptation of LicenseSale.com has been impressive, with work coming from leading medtech companies around the world. The most recent new clients joining LicenseSale.com include T-Doc Laborie from Canada, Seabard from the UK, Luminous from Israel, Mitsui from Japan, Biomeria from France, and Amico from Egypt. Our service model is based on a fixed fee per application multiplied by the number of applications. Typically, an application covers a single family of products registered in a single market. LicenseSale.com offers capabilities and advantages that affect all aspects of the organization. We refer to this as knowledge above the cloud. When using LicenseSale.com, the legal department regains control of the intellectual property. It releases the VP of Sales from his dependency on local distributors. The business development manager can gain market access to any market in advance without the need to have any presence or investment made in the potential new market. It allows the head of regulatory affairs to become a strategic, proactive division of sales and marketing and to finally have control of the registration process in each and every market throughout the complete regulatory life cycle. And for the CEO, all of this translates into growth, revenue, market share, profit margins, and stakeholder interest. Our technology is the driving force behind a revolutionary product which is changing global licensing and registration with an integrated strategy and the tool to streamline, simplify, and expedite your regulatory process. If you'd like to learn more about how you can harness the power of the cloud as a strategic force in your global go-to-market strategy, visit us online at licensesale.com. And now I will pass it over to my colleague John Iannone from Toxicon.
Thank you, Mark. I hope everyone can see the start of the second half of the presentation. Here we'll be transitioning from how you can look at the regulatory process in getting your product out to market and take a step back and before filling into that regulatory process, look at the spectrum of testing that is required for medical devices that utilize colorants. And before we get into that, I just want to give a quick overview of the second half of this webinar. Just give you a quick company introduction. We've already done that mostly for Toxicon. But then talk about why do we use colorants in medical devices. Oftentimes, a lot of people are coming to the stage with these devices, and oftentimes they're saying, well, you know, maybe it's more trouble than it's worth. So we're going to talk a little bit about that. Then go into some of the regulatory testing considerations and come back to, you know, what really is the big deal about colorants. It sounds so trivial, but it's a big impact to the industry. And then finally, we'll go over some options and some ways to meet regulatory expectations. So Toxcon is a contract testing lab headquartered in Bedford, as you've already heard. We have another laboratory in Leuven, Belgium. And for nearly 40 years, we've been assisting the medical device and pharmaceutical industry in 4 key areas: biocompatibility, material characterization, toxicology, and microbiology. We're FDA registered, ISO 17025 accredited, conduct most of our testing to either GLP or GMP compliance, and have over 150 researchers. So with that aside, let's get into the nitty-gritty of colorants in medical devices. There are a lot of different devices out in the industry, and there are a lot of devices that use colorants. Here on the slide, we see different feeding tubes, different tubing, different types of IV lines, catheters, surgical sutures, contact lenses. Colorants are all over in medical device application. So, of course, they've been used in the industry for quite a while. And why do we use them? Well, medical device manufacturers have been using these pigments, colorants, lakes, etc. in medical devices for several reasons. I've outlined a few here. Improvement on ease of use, identification for either product type, size, etc. You can imagine when you have a number of different sutures, having a color additive to help in identification of one suture over the other is definitely a benefit. Also, marketing and visual appeal. Many oftentimes, the product development engineers will come to us, or the program managers will come to us and say, yes, marketing wanted to make sure that this device was red, blue, and green, or whatnot. But it is required for all medical devices to be safe. And I'm sorry, also it's important to look at the ISO 10993 biocompatibility testing, whether we have the colorants present or not. So any change in the additives of a device can really impact biocompatibility, any additive at all, including colorants. And let's kind of go over that real quick. Why is that? Biocompatibility, in its simplest view, is the material being biologically compatible by not eliciting a local or systemic response from a living system or tissue. That means if I put the device in a system, in a person or an animal system, I'm not going to get any unfavorable responses. Essentially, the body doesn't even react as if it's there. That's biocompatibility, not to be confused with efficacy, where we may want to elicit a certain response. From a regulatory viewpoint, biocompatibility is the series of tests that are used to identify potentially adverse reactions from the material or device that's contacting the body. What that means is the leachates of a device or the device itself should not produce adverse local, systemic, tumorigenic, reproductive, or developmental effects. So in biocompatibility, we're looking for a variety of different types of reactions and mechanisms and to ensure that the product is safe. Before we go into those tests, we are taking our device. Maybe it's the small suture. Maybe it's a catheter, segments of a catheter. Maybe it's a very large instrument or diagnostic equipment. So we have to prepare those test articles for testing in the assays. And in biocompatibility testing for regulatory submissions, the test articles are to be finished devices ready for market. That means we have considered all of the manufacturing processes. We are using the devices with the additives that will be present. We have the packaging considered and already incorporated into the process. We have sterilization, a very key aspect, already conducted on the device, so that if there's going to be any effect to the material or the device, that's captured in our test article and then appropriately assessed in our biocompatibility testing. So most tests that are performed for biocompatibility are performed with an extract created from these finished devices. And the extractions are in the test article preparation is commonly utilizing ISO 10993 Part 12 as a guidance on how to do those extractions. There it states that the extractions should occur at the highest feasible temperature and time that the material allows for. One example from the guidance for that would be 70 degrees C for 24 hours. And I'm sure many of you have already seen other extraction durations, 50 degrees C for 72 hours, etc. It also states that you should be using both a polar and non-polar extraction vehicle. This is considering different solubility factors. different compounds that are going to come out of our device and go into the extract solution that subsequently will be used for assessing biocompatibility, well, these different compounds are going to be soluble in different things. We can think of oil and vinegar, oil and water. They don't mix. Well, the compounds that go into the water or the vinegar also will have an affinity to each of those two different types of solutions. So we're utilizing these two different extract conditions to mimic various environments in the body. We have the majority of our body being very polar. Water-soluble type compounds would be appropriately evaluated in that manner. But also we have the non-polar environments. We have our lipids, things of that, fatty tissue, things of that sort. So in biocompatibility testing, according to the ISO 10993, there are a variety of different mechanisms that we would be evaluating for, as I indicated earlier. And all the different parts help identify how to conduct those tests, when to conduct those tests, and we're looking for things like cytotoxicity. Will the extract of my device or the device itself cause cell death? Sensitization. Will the extract cause an allergic potential or an immunogenic response? Irritation, systemic toxicity, both from short-term and long-term exposures, genotoxicity. So you can see here, what we're really focusing on is what happens. Does my extract elicit one of these types of unfavorable responses? And if so, to what degree? Well, if we take a step back and we start thinking about the additives in the device and how those extractions are modeling the device in the body and then therefore assessed in the biological reactivity testing, what we have are all of these additives that are within the polymer. And if you can see here, I don't know if you can see where I'm pointing to, but all those wavy lines, that's my polymer. That's my plastic material, if you will. And I have all these little red dots that are the different additives. And when I expose them to an extraction solution with extraction conditions, which is seen by the blue area, that is my water, so to speak, then what happens is there's a transfer of those different additives from the polymer to the extract solution. And that is why I can then use my extract solution to assess for biocompatibility, the different mechanisms. But what if I were to look more specifically at what those additives are? Here we can start to look more specifically at what is the cause of all of these different types of reactions that we mentioned in our biocompatibility testing. So let's bring that all together, biocompatibility and material characterization, which is what I mean when I'm talking about what is the cause. So biocompatibility is using ISO 10993-1, and it is assessing specific mechanisms for those types of reactivities. It's very material focused, and typically they're polymeric devices that are extracted for the assay. If you look at the toxicology of different compounds, different additives, that's where we are looking at the type of effect a chemical compound has. So there we're evaluating the leachates that are found during product use, the device in contact with the patient clinically, and we want to demonstrate safe and allowable limits of these compounds that are delivered to the patient. And how we can model that is with extractable and leachable testing. That's the chemical characterization, material characterization. Here we can determine the leachates, pre and post interactions between material, sterilization, aging, environmental effects, everything that we've talked about that affects extractability, and how we can characterize what may be of a safety concern. Maybe it will help us with our material selection. For the purposes of this presentation, we're talking more for submission support. So what is the big deal about colorants? We've talked a lot about biocompatibility. We've talked about extractable leachables. Why don't we talk about the FDA position on FDA additives for medical devices? Well, if you look at the FDA's position and what they say about colorants that are used in medical devices, there's three big things to talk about here. One, devices containing a color additive are considered unsafe and thereby adulterated. So that's the most important line that I want to point everyone out to in that paragraph. The FDA says if you use a colorant, they already have a stance, they already have a position that there is a strong potential for an issue. They have already noted in previous documents that there is a strong potential for carcinogenicity, mutagenicity from these different color additives. The next point to bring up is unless a color additive and its use conforms to the listing regulations, then they still have a concern. And what they have identified is the majority of the color additives for medical devices are usually focusing on contact lenses and sutures. Not exclusively, but I will say the majority of what I've noticed through the approval of different colorants, the specific use is, again, mostly contact lenses and sutures. So if your device is a contact lens or a suture, you have a nice long list of different color additives to look at. If you have an IV catheter, your list may be very short or maybe not applicable at all to your device. And then finally, where do we find a lot of this information for the different types of additives that are considered safe or unsafe? Well, that's in the 21 CFRs. Most specifically, I want to direct you to parts 70, 71, 73, 74, and 80. Through all of those different parts, you can get a better feel for the FDA position on color additives, not only for medical devices but other applications. But here for this talk, we'll speak specifically on medical devices. So continuing with that, we were talking a lot about biocompatibility. We were talking about ISO 10993. Well, the FDA has their own guidance on how to utilize the ISO 10993 tests, the guidances, and determining what testing is required for your device. And what I want to point out is the draft guidance document that was issued just over a year ago now, in April of last year. Here, there's a section in Section 7 that they speak specifically to the assessment of known or potentially toxic chemical entities. And in that section, I've actually taken the very first paragraph here. And in that very first paragraph, the example that they use is color additives. As you can see, right away, within the second line, you see color additives. So the FDA's position on color additives and colorants is that any device that uses them is most likely adulterated and that they are potentially of toxic consequence. So when we're looking at medical devices and the composition of these devices and commercial polymers, we have all these different types of extractable leachable sources, additives, residues, oligomers, degradation compounds, both from the polymers and the polymer additives. And if we look specifically at the additives, you can see there's a lot of different things that make my device behave the way I want it to in order to have the properties that I need and be able to withstand certain conditions. The processing for my medical device during manufacturing. But here I have outlined in red the pigments and colorants is one of those sources. So if we look at that, we already know what they're there for. They're to give a certain desired color. They could be also for some type of opacity. They could be there for identification, like we talked about earlier. So what is it that's giving that? It's not that I put like red food coloring into my device to get a certain color of the device. I have different chemical entities that result in my colorant. And this is important to understand. So all of these compounds, pigment green 7, solvent green 3, solvent yellow 114, carbon black, these are all derived from certain chemical compounds. As you see, the PNAs from carbon black, known by many toxicologists to have issues. Other compounds that have less of an issue. But all of these colorants have unique formulations in order to have the specific color and the specific compatibility with the base polymer that you need for your medical device to incorporate these colorants. And this is where the FDA is looking for the medical device manufacturer to better understand what is present in their device and what could be potentially leaching off. So when I say leaching off, we have our polymer represented again by wavy lines. Here I have a more semi-crystalline polymer. And all the different additives. I have these red dots, these green dots, polymer additive fragments. And through diffusion, whether it be from clinical use, whether it be exposure to a solution like delivery of saline, or a drug product, or just direct contact with blood within the circulatory system, that diffusion then allows for these different additives to diffuse and be exposed to the patient. And it is here that the FDA is asking for the medical device manufacturers to understand the impact specifically from colorants and color additives. So here we go back to what is the cause? What could cause systemic toxicity? What could cause hemolysis or whatnot? In order to do that, we want to better understand extractables and leachables. I know we've talked about it, but why don't we talk a little bit more about what the sources are of these leachates? We talked about the different polymer additives. Again, I've outlined colorants. There are all these different degradation products, impurities, migration from secondary contacting material and packaging. All of these things make your extract solution very complex to analyze to look for a specific chemical entity like the colorants. And that really is the focus when you're looking at the chemical characterization with an endpoint of evaluating the toxic potential of colorants. And in order to do that, we need to understand what we're looking for. There are a variety of different organic compounds that may be of concern from all these different leachable sources that we indicated. There are also some inorganic compounds. Many, many pigments and inks are metal-based complexes, like we saw in the structures earlier. When we had the solvent green 7, we saw that there were, if you noticed on the structure, a lot of these chlorine pieces to the colorant itself. And that would be best found by a method that can look and detect these different inorganic compounds. Here we would use ICP. For the various different organic compounds of concern, like we have up top, the volatile organic compounds, the semi-volatile organic compounds, even the non-volatile organic compounds. These compounds and any colorants that fall into these categories would need a different method, perhaps something like GCMS or LCMS. And if you look for those compounds, then that's when you'll find it. But if you don't look for it, you won't find it. And anyone who's seen a presentation from me before knows that this is a very important concept. The more information you know about your colorant, then the more you can go focused in your evaluation and analytical exercise of detecting those compounds. But the less you know, the more you need to do this kind of detective work of seeing where is it, where are the different breakdown products of that colorant perhaps, or the colorant itself. So how are we able to detect these different compounds? Of course, the more we know about the chemical compound, the more we can zero in on the compound attributed to that colorant. But if you know less and less, which seems to be the case a lot in the industry, you want to use some general screening methodologies, first pass testing for extractables. And you use the different instruments, like I noted before, Headspace GCMS or Purge and Trap GCMS, other GCMS methods, LCMS, ICP. Those are more focused on colorants. And you want to leverage a compound database that allows for a more thorough evaluation in a first pass screening. That allows you to use standard methods combined with targeted analysis. The more we know about the colorant, the more we can start to try to fine-tune things. But with these standard screening methods, it allows you to be more efficient and cost-effective in your first pass experiments. So what are we trying to do? We're trying to understand what's happening. Extractable testing, we're going to identify potential compounds that extract into model solvents under more aggressive conditions, similar to what we saw in the 10993 Part 12. And there's where we'll determine which, if any, compounds may affect the patient or maybe the product if we're delivering a drug product. Here we're focusing on what may be present. In the leachable testing, if we need to further evaluate exactly how much is leaching off, what's the elution profile, then we want to measure the actual amount of a target compound. Maybe it's our specific colorant or a breakdown product of that colorant that a toxicologist may have determined as of critical concern, and then determine how much of what is present so that you can do that tox assessment and evaluate the exposure of that product and whether or not it will affect patient safety. So that leads us into risk assessments. And I'm going to talk about two complementary but not overlapping areas that sponsors really should be thinking about in terms of risk assessments. In 10993-1, the guidance that helps outline what testing is required really is through a risk management process. And you have to understand your device, your application, and how that will suit the needs of determining safety of the product. If the biological reactivity testing that's outlined in Part 1 through these different tables, flowcharts, matrices isn't adequate, then the guidance document, if we go back to the guidance document that the FDA issued as a draft last year, it actually says, look more at what you have and understand if you need to do more testing. Understand if the biocompatibility testing that is outlined in these tables is truly sufficient and adequate. And they bring up that when they're talking also about colorants. The next piece is the ISO 10993-17. And here we're establishing allowable limits for leachable substances. These leachable substances can be all sorts of different additives like we talked about, but more specifically for this presentation, it could be our colorants. So the Part 1, as I mentioned, if you want to evaluate the device materials, perhaps a component breakdown, consider history of clinical use, and identify the data gaps. This really should be performed at the initiation of your safety testing. And the Part 17 really should be used more as a follow-up. A follow-up to what? Well, it can be for the biocompatibility testing. I think that it's most valuable when we're looking specifically at the extractables and leachables testing that falls in the 10993-18. Here we want to review safety information for those identified compounds, set limits of safe exposure, and compare those limits to the amounts that we found in our elution testing. And really here, not only do we get a green light from continued product development, but we also have more support for our regulatory submission. Now, a lot of people ask when we're talking about colorants, well, how do I get out of having to do all this extensive testing? I mentioned earlier that there are some colorants that are approved specifically for medical devices. I really want everyone to understand how short of a list that really is. Of course, you can't really read everything there, and we can share some of this information after the talk. But you can see here the colorants that are approved for specific uses. Okay, there are use restrictions, but for specific uses, like we talked earlier about for different contact lens applications or sutures. The ones that require batch certification, I don't know, that can't be more than 10 compounds there. In the next slide, yes, we have more colorants that are approved, again, for specific uses. We have a whole bunch here. There's about 6 compounds, right, colorants that are approved right in the middle of that table, and we can see that's all for contact lenses. As a matter of fact, the second half of this entire table is specific to contact lenses. And even above that, we can see a number of compounds that are batch exempt or batch certification exempt and approved for contact lens application. So if we're trying to understand how do we move forward for our assessment of colorants in medical devices, we've talked about the use of chemical characterization to understand what is leaching. We talked about biocompatibility testing to understand what kind of an effect we have. I've outlined a few options here that we can consider for meeting these regulatory expectations. Let's take the example of a device that has various different configurations. And when I say different configurations, maybe it comes in blue, red, and purple. Well, you can conduct your full biocompatibility on each device configuration because the one that's red and the one that's blue and the one that's yellow, they're not all the same because of that colorant, that chemical compound, and then have a technical evaluation of each colorant. Now, I'm not going to say that that's the most effective way to do this. I'm just saying that that's one of the options that could meet your regulatory expectations for your device. The next one is to do full biocompatibility on one device configuration and chemical characterization on each device configuration. That would be each of the different colors that my device comes in. And then do a toxicological risk assessment of the full package of the biocompatibility results, the chemical characterization data, trying to correlate pieces of this. And that's the second option. Another one would be to conduct full biocompatibility on one device configuration and conduct a toxicological risk assessment of the additives. And here we're focusing on colorants. So we could do, if we have good information from the suppliers, if we know the colorant structure, the chemical structure of the colorant, if we know more about the toxicity of these chemical compounds, and the elution profile, or assume that everything comes out of the device. All of the colorant that is added all comes out of the device, and whether or not we still have any kind of toxic concern, that can also be evaluated as an option. And the last one is maybe to conduct a pooling of all the different multiple device configurations and conduct the biocompatibility on that pooled extract soup, if you will. And here we would need to modify our extraction ratio to maintain a certain level of aggressiveness, so it's protective of the use of the product and ensure that the individual configurations are not diluted with the amount of extract solution from other components and configurations. So instead of taking 4 configurations and doing an extraction that we use 100 mLs for the entire 4, because that's what the surface area to extraction ratio would mandate, using a 3 centimeters squared per ml extraction ratio. These are all from the guidance. Maybe we need to have those 4 different configurations and extract them all within 25 mL, so that we have the same concentration of each individual additive that could be unique to each configuration, and we don't dilute them out. What's the risk in doing this? Well, the other risk is that any common chemical entities from the 4 different configurations is now intensified in our biocompatibility testing. But that, along with a technical evaluation of each colorant, can still be a suitable approach for evaluating the safety of that product. And that really comes down to understanding whether or not the biocompatibility testing is going to be sensitive enough to evaluate any effect from my colorant. One thing that I'll say before moving on from this slide is we've seen this area, and the reason why there's been such a large response to this webinar, we've seen this area blow up. A lot of people are asking, why am I getting these deficiencies? Why is the FDA asking me all these questions about colorants? Well, the fact is, in many biocompatibility tests, there are chemical entities attributed to colorants that will not give you a response, but is a concern. And there have been product recalls and different types of feeding tubes that have been pulled on the market because of the use of a colorant that was not adequately assessed, but did have the appropriate biocompatibility. So the FDA is now saying, listen, we want you to look at this. This is a concern. We have the safety of the population in mind, and we need you to do a thorough investigation whenever you use a colorant. And I think that sounds daunting, but it's actually very achievable. And as we mentioned earlier, through a variety of different techniques, there is ways to gather the right information and assess the overall safety impact. So in conclusion, I just want to say that colorants, inks, and pigments are considered additives to medical devices, not just food coloring. And understanding the full characterization of these additives through identification, elution characterization, and patient safety determination will eventually satisfy the regulatory expectations and requirements. Biocompatibility may be leveraged along with chemical characterization to evaluate more than one device configuration when only a single difference exists. And that's our example of the same device that comes in 3 different colors. And the toxicological risk assessment of device additives can be sufficient with good information and exaggerated conditions of use. And I will say, when it comes to colorants, that tox risk assessment, those technical assessments of those chemical entities are the most critical aspects to making sure that you have all the information together to determine if the use of a colorant in your device is truly going to cause any kind of a safety concern. And with that, I want to thank everybody for listening in on the 2 presentations, and certainly pass this over for our Q&A session.
Great. Well, thank you very much, John, for that, and thank you also to Mark for those very insightful presentations there. At this point, I would like to invite our audience to continue sending their questions and comments, and right now using that questions window for the Q&A portion of the webinar. And gentlemen, I've already received some questions, so I'll start with the ones that we have here. Our first question, what if new requirements are implemented when we are registering a device?
Hi, thank you for that, whoever submitted that. What we do at Erasmus Group is we work to stay aware and ahead of regulatory changes as they come up, and that includes all countries around the globe. If a change is in fact introduced, then what we do is we generally add those requirements to the list of documents that are in the client portal, and that way the client is aware of what those requirements are right away. So it will be visible to them when they actually sign into the portal and are accessing their documents.
Great, thank you for that. Our next question here. If I have conducted all my biocompatibility testing on my device, do I need to do extra testing for colorants as well?
Yeah, that's actually a good question as well. So as we indicated in the presentation, there are instances where biocompatibility testing really isn't sensitive enough to always evaluate the potential safety impact of the colorant being present in the device. Some degree of evaluation is required, especially in the current landscape of the FDA regulatory submission process. What I would suggest there is first to evaluate the colorants, and if you have information about the colorant additive in terms of a chemical structure, to evaluate what is the potential impact of if all of that product or that chemical compound comes out. And if there is still a concern, then it's going to be critical to evaluate the actual elution profile so you know how much of that compound comes out, and you can then determine what is the safety impact of that level of that colorant coming out and being exposed to the patient.
All right, thank you for that. Our next question here, what specific types of solvents does Toxicon use when performing leachable or extractable testing of colorants in plastic materials?
That's also a very good question. So the use of solvents is a non-trivial aspect to extractive leachable testing. It's a critical part, as we can see from the presentation earlier, to mimicking certain types of environmental effects. It's important to understand what are we trying to model in order to determine what is the most appropriate solvent. Of course, if we're trying to do this in a very generalized manner, the guidances talk about polar and non-polar for medical device applications, and we can do that with the use of purified water and something like hexane. If either one of those solutions are not compatible with the material, just like in the biocompatibility testing, if a certain condition is not compatible with the material, then we do need to look at alternatives. It's important also to consider the use of the device. So when I say polar and non-polar, well, if we have a device that's in direct contact with blood, there is some acceptance and some guidance to using an alcohol mixture of a lower concentration in order to mimic that type of a condition. The key in solvent selection is understanding what are you trying to model and what is the intent. Are we trying to be aggressive in terms of extractable testing, or are we trying to be more simulated use, like in leachable testing? And then finally, make sure we have good material compatibility. I think that answers the question there.
I think so. Thank you. Our next question in a couple parts here. How is bioavailability defined? Is it the same as leachability? And how is leachability evaluated as compared to extractability per part 12?
That's a great question. So bioavailability, in its simplest way, we're trying to see what becomes available to the patient's biological system. Leachability is what is coming off of the device under clinical use or simulated use. So the leachability looks at factors like diffusion, solubility in the conditions, and molecular weight of the compound, etc. Extractability is whether or not we can pull that compound out under more aggressive conditions for identification purposes, but not necessarily look at that as the total amount that will be delivered to the patient, which is then related to leachability and bioavailability. So of course it's really important, and this is why it's such a great question, it's really important to understand all three of these mechanisms on how they come together. But yes, they are all slightly different. I would say it has to do a lot with from what aspect you're looking at. Leachability from the device under clinical use, or bioavailability from to the patient in terms of toxicological evaluation. And of course, extractability in order to better define what you have to start worrying about in the first place.
Right. Okay. Thank you for that. Our next question here. The FDA also states that this provision applies to color additives that directly contact the body for a significant period of time. What has been the regulatory experience with the definition of significant period of time? And is this implant only?
Also a good question. When we're talking about colorants and better characterizing and evaluating the impact of a colorant from a device, certainly the longer of a contact the device has with the patient, the more of an impact it will have in terms of leachability and then therefore bioavailability, going back to the question previous. Now, we have seen, even with limited exposure devices, that sometimes be a concern. It's usually more attributed to if there's some type of a delivery of a drug product or a solution for limited exposure. But anything that is defined as prolonged exposure or permanent exposure would then really necessitate an evaluation of the impact of colorants. I will say that that is not unique to only implants. The guidances actually break out three main categories: surface contacting devices, external communicating devices, and implantable devices. Each one of those have their three different durations of exposure. So for the short-term contact, which is less than 24 hours, I'm going to say there are some unique instances when we need to worry about colorants. But then when we go into the prolonged exposure and permanent exposure, it's really focusing predominantly on the external communicating and implantable devices, but not unique only to them.
All right, thank you for that. Another question here. If a pigment or colorant from 21 CFR Part 73 Subpart D is picked, this was referred to on slide 12, I believe, would that require less biocompatibility testing? This person goes on to say, I did not understand the last bullet point of slide 12. If you could review that for us a little bit.
So could you just repeat the question one more time?
Sure, absolutely. If a pigment or colorant from 21 CFR Part 73 Subpart D is picked, would that require less biocompatibility testing?
Okay. So if you were to choose a colorant that has been approved for a specific application, then the colorant itself is approved for that specific application, but the other additives, the other degradation compounds from the polymer, all of that still will require the assessment of that device for its application. So the biocompatibility that is required for a medical device, I'm not going to say if you pick a certain colorant that is approved for a specific application that it will minimize your biocompatibility. But certainly it may minimize the additional testing that you need to do specific to the colorant. The medical device has its own set of requirements, and that has everything to do with the polymer that's in the device or the different types of manufacturing processes, the additives, all of those types of other leachable and extractable sources that can influence biocompatibility. So it's also a good question, but it's very important to keep apart what is required because a colorant is present and what is required because you have a medical device.
All right, thank you for that. Our next question here. What are some of the recommendations for testing metal devices for short-term skin contact? So
in terms of biocompatibility for a metal device with short-term skin contact, if there is a conformance and you can confirm that conformance to a specific standard, like different ASTM standards for stainless steels. If you have the ability to conform to that standard and confirm it, then the biocompatibility of that device is defined by the conformance of the standard in and of itself. And that happens quite often with some types of metal devices. However, if we have a metallic device and we can't claim conformance with a specific ASTM standard for that material, then you don't look at the fact that it's metal, but you look at the application of that device. Going back to before what I indicated, different types of patient exposure, surface contacting, external communicating implantable, they all have their own biocompatibility testing. I think the question here was for a limited contact surface device. Is that correct, Andrew?
Yeah, that's right.
Yeah, so for a limited contact surface device, aside from the fact that it was a metal or polymer or whatnot, that is going to require the cytotoxicity, the sensitization, irritation evaluation. So that is, again, specific to the device application, not the material of construction.
All right, thank you for that. Our next question, this audience member says, if I have a raw material declaration that the colorant has already been tested to ISO 10993, does that help?
Well, it certainly helps to have some raw material testing, but when you take that raw material and you process it, and you form your device, and you may add other additives, and you do a certain sterilization process, the extractability, the leachability, even the compounds that will come out could potentially all change. Does it help to have some initial medical material qualification type testing? Absolutely. It means that you're not starting from scratch in terms of the relative safety of the product, but try to understand a raw material resin versus a final finished device. That's why in the 10993 Part 12 they talk about testing the final finished device, because all of these other steps that we go through in making a device can influence what is coming off and eventually delivered to the patient. One other thing I want to bring up is when someone says conformance to the ISO 10993, just try to make sure you understand what that is. Because 10993 is not a specific prescription of testing. There is a matrix of tests to conduct and different mechanisms to evaluate for, depending on the device patient contact and duration of contact. So the question earlier about a surface contacting device for limited exposure, the biocompatibility according to 10993 is very different than, say, a permanent exposure or a greater than 30-day exposure of a catheter system to the circulatory system of a patient. The biocompatibility testing would be very different.
Right. Okay. Thank you very much for that. Our next question here. How can you do a risk assessment when the dye manufacturers refuse to disclose the identity of the colorants?
That's probably one of the best questions today. I'm actually going to, I love this question so much that I'm going to tell you a little story. I was on the phone with a client and the FDA supporting them with their regulatory submission with the testing that Toxicon had conducted and explaining what are the pros and cons of everything that we did and what were the limitations. And in that discussion, the FDA was absolutely asking repeatedly just that question. You know, what are the chemical entities of the colorant? Can you identify what extractables and leachables were identified are attributed specifically to the colorants? What are the chemical structures of the colorant? And of course, with less and less participation or cooperation from the colorant supplier, there was less and less information that was able to be attributed specifically to that colorant. And the FDA, during that call, actually said to the medical device manufacturer, give us the name of your contact. And I'm not saying this happens every day, but they said, give us the name of your contact, and we will contact them and say that on behalf of the FDA, we want to know what the chemical structure is. So yes, I agree. It is very difficult to do a risk assessment on a color chemical entity if you don't know what the color chemical entity is. And this is why a relationship with the colorant manufacturer is very critical to getting that kind of information. But when you do not have that information, your only best bet, unless the FDA is willing to make a call for you, your only best bet is to take and isolate resin with and without colorant, device with and without colorant, and isolate them and do the chemical analysis, leveraging a very strong and powerful database to be able to detect and identify different chemical entities, and hopefully through an evaluation of the delta with and without the colorant on different test articles, you can start to identify the source or potential sources to the colorants and which chemical entities are responsible for them.
All right. Interesting. Thank you for that. Our next question here. Can you address security issues with a cloud-based service?
Sure. Let me speak to that. First of all, we use Salesforce Cloud, and all the security issues are covered by Salesforce as part of their global cloud services. And Salesforce and Oracle Clouds were recently merged, which provides one of the world's stronger and more secure clouds that are available out there today. So we've certainly covered off all the issues involving security to our clients and use of the portal through the cloud.
Great. Thank you, Mark, for that. Our next question, does the use of colorants in plastic consumables for packaging, be that final and intermediate, of pharmaceuticals and biologics raise the same concern as devices?
The FDA has also looked at the use of colorants in pharmaceutical packaging material, and there is another list altogether specific to the use of colorants that are approved for specific applications for drugs and pharmaceuticals. I didn't focus that here, but a lot of the mentality of the FDA is similar. Here, the extractable leachable testing is much more involved for a final container closure system or a packaging system, either intermediate or final. So the extractable leachable testing does have to consider the use of colorants, and if the colorants are not approved for that application, then they have to be identified in the leachable study and then be able to determine the safety of the product.
All right. Thank you for that. Our next question. You mentioned feeding tubes, sutures, contact lenses, etc. If conditions of use for a device are more akin to using a ballpoint pen, but only for minutes a day, is it appropriate to reference pigments that meet the requirements of 21 CFR approved for use in food contact applications?
That's a very good question. I don't know if that is exactly appropriate. It would be difficult. If the product is still defined as a medical device, then I would still stick with the medical device approved colorants. Now, if the same colorant is approved for food application, then there may be some way to leverage some of that information, but I don't think going with just it's approved for a food grade application and that's why it's okay for my device would be considered acceptable. I think there needs to be more of a technical evaluation of how the two are associated and how one can still be protective of the other.
All right, thank you for that. Another question here. Will the revised USP 661 subchapters affect currently approved products?
So my understanding of the revision to the 661 is that there isn't going to be a requirement to go back and reapprove materials that have already been tested to 661. In fact, there was a clever statement that was incorporated into the revised 661 that stated any material that has already been approved through the 661 process, by definition, will still be approved for the monograph 661 currently. So they don't say you have to go back and do additional testing. but they do say that if it is approved, it's considered approved if there is any change. Now, that's the other piece to this. In the new 661, there is a much more greater understanding and impact by the different manufacturing processes or whatnot to the material. And they stress the importance that if anything is modified, that you have to requalify. Well, the requalification process then has to adhere to the current 661 expectations, which in fact then would be a requalification of that material. But existing materials with no changes, everything being identical, that by definition, from the current understanding of 661, would be considered approved still.
All right, thank you for that. Another question, is there a specific recommended test for VOC testing for respiratory devices extracted from gas?
So that's a big topic that has been discussed over and over. There is some discussion going on with the important individuals, the critical individuals that are helping to guide the guidance requirements for these types of systems. And there is discussion about what is appropriate, and depending on the different type of respiratory system, then what type of a level is appropriate for evaluation. So there's still work being done here. There is no defined, or to my knowledge, a already defined analysis that is considered appropriate. With that said, there are some ways to capture gases and analyze them through purge and trap GCMS, and that's the direction where a lot of this is going as of a month and a half ago.
Interesting. Okay, thank you for that. Another question here. What if we can't meet the new regulatory requirements? I believe this is a question from Mark.
I'm hesitating because I don't believe that we've ever had that situation. The only scenario that I can think of is that where there might be a new requirement that entails perhaps a clinical or a biocompatibility study. However, that scenario is unlikely, as requirements are generally reflective on the class of medical devices. So in most cases, the studies are automatically included in the requirements of any high-risk devices. For them to be added, lower-risk ones seems pretty unlikely. If it is a case in which the new requirement involves a license that the company can't obtain or is not eligible for, we can follow up with the regulatory body. We do that in the specific country to see if any other documents can be substituted for the requirement. This particular scenario doesn't generally occur, as we're constantly updating requirements and making those changes within the system, licenseheld.com.
Right, great. Okay. Another follow-up for you, actually, Mark. Could you elaborate on the pricing options?
Sure. So pricing depends on a few different factors. First of all, the class of the device, specific country fees, and specific country requirements. For instance, in some countries, you might be required to complete both a company registration and a device registration, and that may result in a few extra pieces of documentation. Also, when you do register your device in multiple countries, you find that it's less expensive in some than in others due to country-specific fees or exchange rates. But based on previous applications of all the clients that we've had over the years and registrations done around the globe, on average, our clients pay about $8,800 US per application. If you decide to register the device through us, you're obviously provided with a quote proposal prior to the process based on your device's classification and the countries that you choose to register in.
All right, thank you for that. Another question here, and this is a follow-up on the solvent for extraction and leachables question. For a combination device drug product, would the best solvent to draw out extractants and potential leachables be the drug solution within the medical device, since the medical device is not in contact with the body?
It's an excellent question. So the selection of an appropriate solvent for an extractable study should not only mimic the contact of concern, in this case would be the drug product to the device, not the patient itself, not only mimic the contact of concern or the environment of concern, but it should also be amenable to the analytical methods. So what I mean here is I want to use a relatively clean model solvent so that I can detect compounds in that solvent. If I have a very complex matrix that I'm analyzing within, then I really have to have a good understanding, or maybe it's impossible to fully understand, whether or not I have any kind of matrix interference, whether I have any kind of coelution of different chemical entities that don't allow me to detect compounds that I was really not ready for. Because in the initial extractable testing, it's a forensic activity. We're going in and trying to see what's there. Well, if we have a complex matrix, the more complex it is, you can think about it more as searching for the needle in a haystack. You may start covering things up and you're not able to detect. So the alternative is to look at these cleaner model solvents and mimic the environment of concern. What I mean by that is if I have a drug product that the different chemical constituents to the formulation can be modeled with, say, a polar extract, an alcohol, perhaps something to model a surfactant within the formulation of the drug product, then I can use these model solvents as building blocks to examine what may be present. Finally, in my leachable study, I'm going to have selected specific chemical entities from my extractable study, maybe from some raw material evaluation, vendor information, and move into my leachable study with target compounds. With these target compounds, I can develop and validate methods that should be able to deal with any kind of matrix interference and then focus my method to be able to overcome those obstacles. So that's why it's really important to not use the drug product right out of the bat, unless you're talking about something super clean and not going to have an issue. But most of the times, most drug formulations are rather complex matrices. So we have to use these model solvents first off to do my forensic activity and identify what I may be concerned with, and then move into my leachable study, which is simulated use. Here, the drug product would be completely appropriate to look for a target compound.
All right. Well, thank you, John, and thank you, Mark, very much for those answers. We have reached the end of the question and answer portion of this webinar. If you do have any further questions, however, you can direct them to John Iannone via the email address showing on your screen right now. Thank you, everyone, for participating in today's conference. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event, and a survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our further webinars. Now, please join us in thanking our speakers for today, Mark Thompson and John Iannone. We hope that you found this conference informative. Have a great day, everyone.